Dear participants, we will be starting the program in few minutes. Thank you for your participation and logging in. So we will be starting the program in short while. Thank you all.
சிவா பெர்ஃபெக்ட்டா வந்து உட்காந்துட்டா, நல்லா நீட்டா. ஹெட்ஃபோன்லாம் சும்மா போட்டு இந்த இளையராஜா ரெக்கார்டிங் மாதிரி இருக்கு
சார். எல்லாரும் இருக்காங்க சார், ஏன் சார் ஓட்டுறீங்க?
அப்படிலாம் இல்லைங்க, உண்மையா சொன்னா என்ன ஓட்டு. யாருக்கு வந்துட்டாச்சா?
ஜாயின் பண்றேன்னு இருக்காரு.
சரி சரி சரி, ரவி வந்துட்டாரு போல இருக்கு.
ரவிக்குமார் சார் வந்துட்டாரு.
சிவா
சார்.
ஃபர்ஸ்ட் ஸ்பீக்கர் முடிஞ்ச பிறகு நீங்க என்ன கால் பண்ண பிறகு நான் வந்து இன்ட்ரோடியூஸ் பண்ணுவேன் இல்லையா குணாலன் சார்?
ஆமா சார், ஆமா சார், ஆமா சார்.
ஓகே, டன் டன். நிறைய ப்ரோக்ராம் அட்டன் பண்ணி இன்னைக்கு நம்மளுதேங்கும் போது அது ஒரு இதா இருக்கு. ஏன்னா எல்லாம் நீட்டா வந்துடும், ஒன்றும் பிரச்சனை இல்லை. ஏன்னா எக்ஸ்பீரியன்ஸ் இருக்கு உங்களுக்கு நிறையா. அதுவும் நம்ம அந்த ரோட்டரி இது வந்து உண்மையில நல்ல எக்ஸ்பீரியன்ஸ், ரிஹர்சல் பார்த்ததெல்லாம்.
I think HOD has joined. Sir, unmute பண்ணிக்கோங்க. Sir, unmute yourself.
I have come already.
I think I have unmuted chief guest also.
Okay.
Janardhanan sir.
Janardhan sir. Sir, good afternoon, sir.
Good afternoon, sir.
No, I was actually trying your number. I forgot.
Okay, sir.
I thought of calling you, but okay, I joined at 2:10 itself.
I
Okay, sir,
have
okay,
I think that Jivachandran is
Okay,
seeing
sir,
for the
okay,
first
sir,
time now.
Yes,
Who is
yes,
actually
sir.
admin?
Sir?
Admin is somebody else, no?
No, myself only, sir. Myself only.
Okay. So you
I
didn't
have
see
made
me
a
earlier.
co-host, sir.
Okay.
No, sir. Just now I am seeing, sir.
Right. That is why, no, I joined it. I was trying. It was muted by
Okay,
you.
okay.
I couldn't
Sir, okay.
call you. Right.
Just now I made you co-host, sir.
Okay. Right.
Sir, we'll start in exactly at 2:30, sir.
No problem.
Okay, sir. So, dear participants, we will be starting the program exactly at 2:30 p.m. Thank you. Sir, welcome, sir. Gunalan, sir.
Good afternoon.
Sir. Abhijit, sir. Welcome, sir. Good afternoon, ma'am.
Jayarajan, what is the actual sequence of events?
Sir, சொல்லுங்க sir.
What is the sequence of events? என்ன எப்படி
இப்ப first inauguration முடிச்சிரோம் sir. முடிச்சிட்டு அவர் டாக்டர் அபிஜித் அவரோட டாக்.
இல்ல இனாகுரேஷன்ல வந்து நீங்க எப்படி சீக்வென்ஸ்னு கேக்குறேன் நான். இனாகுரேஷனுக்கு ஒரு சீக்வென்ஸ் வச்சிருக்கீங்களா? அது ஏதாவது இருக்கா? இல்ல ஸ்ட்ரைட்டாவே நான் இனாகுரேட் பண்ணிடுவேனா?
அங்க இது ஒரு சின்ன பிரேயர்.
வெல்கம் அட்ரஸ் சார்.
வெல்கம் அட்ரஸ் முடிச்சிட்டு
வெல்கம் அட்ரஸ். ஓகே. சரி.
இன்வோகேஷன் வெல்கம் அட்ரஸ்.
Invocation, welcome address, and then inaugural address. Okay. Fine.
Sivachandran.
Ma'am, good afternoon, ma'am.
All the best wishes. Do well.
Thank you, ma'am.
God bless you. Okay.
Thank you, ma'am.
Good afternoon, Janathanam sir.
Good afternoon, ma'am. How are you?
Fine, sir. Doing good.
Sir, Tyagran sir, start பண்ணலாமா?
ஸ்டார்ட் பண்ணலாம் சார்.
Yeah.
Okay, sir. A very warm good afternoon to everyone. We start the program with college prayer. Sir Rajagopal sir.
Yeah.
Yeah.
Om Sahana
Bhavatu
Sahana Bhunattu
Sahaviryam Karavavahe
Tejasvinavi Om Shanti 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 Pitasi
Lokasya
Charatarasya
Namasya Pujyasya
Namaskaruya
Gururgaryan Natvassamostyabhyadigatputonyo
Lokatraya
Lokatrayepya Pratima Prabhava
Tasmana Pranatha Pranikayakayam Prasadaye
Tasmadayamahamishamidhyam Piteva Putrasya Sakya Satyuhu
Priya
Priyaya Hasideva Sodam Asato Ma Satgamaya
Namasto Ma Jyotirgamaya
Jyotirgamaya
Pratyoma Amritam Gamaya
Amritam
Om
Gamaya
Shanti Shanti
Shanti Shanti
Sabhagaya Dharmasya Sarva
Sarvadharmaswarupine Avatara Varishtaya
Ramakrishnaya
Te Namaha
Namaha Jananim Saradam Devim
Ramakrishnam Jagadgurum Padapatne Tayoshritva Pranamami Muhurmuhu Namastriyati Rajaya
Vivekananda Suraye Satchit Sugaswarupaya Swami Netapaharine Hari Om.
Thank you, sir. So, Vanakkam, Namaskar. At the lotus fetus of Holy Trinity, with the blessings and guidance of Secretary Maharaj, Swami Sukadevananda ji, we are to start our webinar, a national webinar on scope, employment opportunities, and future research in applied zoology. So, good afternoon to one and all present here. We, the Department of Zoology, Ramakrishna Mission, Vivekananda College, cordially invite you to the National Webinar Lecture Series on Scope, Employment Opportunities, and Future Research in Applied Zoology. Let me take this opportunity to highlight a few elements that shape the webinar to be more constructive for the benefit of students commonly across the globe. So as in Tamil literature, one of the important epics is Mani Megalai. It talks about not only disaster management, but as well as cultivates the nature of humanitarianism among every individual. In this context, the current COVID crisis has given us opportunities to build new strength and new avenues of performance to a larger extent in virtual mode. The Department of Zoology has envisioned the new avenues of employment opportunities in applied zoology that emerge at the current moment, and we like to share such important knowledge through renowned experts in the field of applied zoology in terms of scope, employment opportunities, and future research to youth across the nation. This webinar lecture series will provide a platform to the participants to learn and explore in the field of Applied Zoology. Thank you. Now I invite Dr. Tyagarajan Raman, Assistant Professor and Head Department of Zoology, Ramakrishna Mission Vivekananda College, to deliver the welcome address and introduce the chief guest to the August gathering.
Good afternoon to all. Myself, Dr. Tyagarajan Raman, on behalf of the management, principal, and staff members of Ramakrishna Mission Vivekananda College, would like to extend a warm welcome to all the participants for the national level webinar lecture series on scope, employment opportunities, and future research in applied zoology. I extend my sincere welcome to all the 4 speakers for the webinar series who have given us their concern and their precious time to share with us, as well as all our students, the scope and research opportunities in zoology. Dear sirs, I welcome all of you for this webinar series. Today's chief guest for the day is Dr. S. Janardhanan, who is Professor and Head, Department of Zoology, University of Madras. I have known Janardhanan sir since my MSc days from Loyola College, when he was working as a scientist in Entomology Research Institute. And we had discussed a couple of times there. And subsequently, when I moved over to University of Madras for my PhD, I have known sir for quite a long time since then. Dr. S. Janardhanan finished his PhD from Bharatidasan University. And then he did his postdoctoral fellowship from United States of America, and subsequently he moved over to Tyagarajan College in Madurai, and then he joined University of Madras as associate professor. Now he has taken he's in charge of the Department of Zoology for the last 6 years, and he has published a number of research papers on insect protein, and he has guided a number of PhD students also. Dear sir, I welcome you to inaugurate this webinar series, and thank you, sir. And I hope that your experience will be a big boost for us in our endeavor. Welcome, sir. Thank you. Now I request you to kindly give the inaugural address.
Thank you, Dr. Tyagarajan, for
thank you, sir.
Kindly consented to me, the chief guest of this webinar, actually on a lecture series arranged by the Department of Zoology, Vivekananda College. Thank you very much once again, and I also extend my thanks to Dr. Sivachandran as well as the principal, who has kindly consented to me as the chief guest for the inaugural function. And it is the right time, especially for the zoology students. A very apt title is given as a lecture series: Scope, Employment Opportunities and Future Research in Applied Biology, Applied Zoology, where of course I will be discussing with you all, especially on 2 major areas. One is, what is the scope that you have for research after you do your post-graduation? Of course, postgraduate study is very important to anyone to pursue research in any discipline. And I will be discussing what is the scope which is available, or opportunities which is available for one to undergo research as career. And on the other side, I will be discussing with the teaching opportunities, as well as what are all the other employment opportunities available with several different public and private sector undertakings in India, as well as with other avenues for zoology students to get themselves a career in their life. So with this, I just would like to highlight some aspects of research opportunities for zoology students. First and foremost, when you pursue your MSc, you complete your MSc, when people join for MSc program or PG program with university department, I always advise them to train themselves during the course of the study in the 2 year study, primarily based on the syllabi provided by UGC CSER. So once they come across the difficulties of the subjects in life sciences under UGC CSER, they will be very well compete themselves to attend for the national eligibility test for UGC CSER. There are at least 1,000 fellowships are given both by CSER and UGC as a junior research fellow, and those who have qualified in these UGC CSER net examinations, they will also be qualified for lecturership. And the amount of fellowship, and this kind of fellowship is for 5 years, and you all know that the amount of fellowship, including HRA, will be around 40,000 rupees per month. So that would be more than sufficient for a research student to sustain themselves, as well as to some extent sustain their family also. So it is my humble request to all you students who are having an aptitude for research, you will try UGC CSER. And secondly, there are of course several research fellowships which are given by UGC to all university departments. In the departments which are getting some kind of special assistance program, we call it as a SAP program. And people who get SAP programs in university department, at least 5 to 10 number of fellowships are given by UGC every year. So this is another boost for students who do research in university departments. And of course, you have a lot of sponsored research projects in which you can also get yourself appointed as a project fellow or junior research fellowship or sometimes a senior research fellowship. That will definitely help you also to earn some money as well as do your research other than the area in which you sometimes work in the project or with the consent of the principal investigator. You can also do the same project for your PhD program. So that again varies. So there are a lot of sponsored research projects. And for the SCST students, privileged students, then there is a fellowship from UGC which is called as Rajiv Gandhi National Fellowship. And there are of course fellowships instituted by the Department of Science and Technology through what is called as an Inspire Fellowship. And this fellowship is directly given to students who come as a first rank holder in university examination. So a candidate who has come as a university rank holder will be definitely given without any other merit except the rank certificate. So that is again another very fascinating idea proposed by the MHRD, wherein which DST is instituted such kind of fellowship programs. And you have also the fellowships given by many foreign funding agencies through some nodal agencies in India. To cite a few, there is a Mary Curie Fellowship for primarily you do a research on cancer, and there are also fellowships which are instituted again in UGC for Muslim students, minority students, which is otherwise called Maulana Azad National Fellowship. And in addition to that, very recently in the last 4 or 5 years, there is also a research fellowship which is actually instituted in the name of Prime Minister. So Prime Minister research fellowships are available, which is primarily given to students who join for research program in premier institutions in India like IIT, ISER, IIC, etc. So once you complete your postgraduate program, there are opportunities for you to attend the a kind of an entrance examination to get yourself fit into a PhD program in premier institutions like IIT, IICs. Once you join, there are a lot of avenues for you to get this kind of Prime Minister Research Fellowship. And if you see the support, the extent of support which is given through this Prime Minister Research Fellowship is about 6.1 lakh per annum. Then you can imagine. So there are about 100 fellowships per year is given for this Prime Minister Research Fellowships. So you have a lot of opportunities once you do your PG programs for doing research. So biology students, especially zoology students, they will have a lot of scope. So this is one part of course, the scope which is available for future research. Of course, it may be an applied zoological research or maybe a basic zoological research. But when you have some aptitude for research, you have a lot of avenues. I'm not going to discuss anything about the areas, applied areas in which, of course, you have to pursue in future. That is, of course, secondary. I always consider, you should have real research aptitude. So that alone will be sufficient for you to get a lot of opportunities in research. Okay. And if you see on the other side, and other employment avenues for both undergraduate as well as postgraduate zoology students, you can very well do B.Ed. Subsequently, of course, M.Ed if you wish, and you will have ample number of opportunities both in the state as well as the central governments for teaching schools up to the higher secondary level. So, if you have a kind of a teaching aptitude again, some students they will be very good in teaching. So those students, I always advise better you select whether you are going to be a teacher in a school or going to be a professor in a university or college system. So B.Ed, M.Ed will definitely give you based on your, of course, the societal background, family background. You can do B.Ed and also pursue M.Ed, and you can very well be placed as teachers in schools of both the state and the central governments. And as I already told you, if you have qualified for NET, National Eligibility Test for lecturership, you have also opportunities to serve as assistant professors in colleges as well as universities, even without PhDs. And they give some grace time for about 3 years time. Once you join, then you can pursue your PhD program. So these are all other avenues, especially available for teaching to the students who are actually interested in teaching. So these are the two major areas by which one can always see a lot of opportunities. And again, there are other opportunities like employment opportunities like UPSC and the Staff Selection Commission, Tamil Nadu Public Service Commission, Group 1 services especially. I will say always you attempt for the higher level of positions in these recruitment boards. So Group 1 services, you have a lot of opportunities for IAS, Indian Administrative Service, Indian Foreign Service, Indian Postal Service, and especially for biology students and zoology students, there is a higher level officer examination through UPSC, which is an Indian Forest Service. Indian Forest Service is exclusively for the biology students. One can always have an attempt to, even after your undergraduate program, you can very well write the Indian Forest Service. So this is in the other side, the government employment at a higher level, at an officer level. And thirdly, you have a lot of opportunities in the case of public sector as well as private sectors. And in both the sectors, of course, in the name of what is called as a conservation biologist, behavioral biologist, wildlife biologist, zoo curator, director, wildlife warden, wildlife educator or manager, zoologist, fish and game warden, ecologist, environmental biologist, animal nutritionist, marine biologist, environmental consultants. So there are plenty of opportunities, both in the private as well as the public sectors, where there are a lot of zoological parks, museums, national museums, regional museums, there are national parks, and there are wildlife career opportunities. So these are all a lot of avenues, both at the government side as well as at the non-governmental side. And other than that, India has instituted long back institutes like CSIR, Council of Scientific and Industrial Research, ICAR, Indian Council for Agriculture Research, Indian Council for Medical Research, DBT, DST, and the Zoological Survey of India. So these are all various public funded organizations instituted by various national agencies and the government of India. You have a lot of opportunities from a lower grade as research scientist to a level of research assistant to the level of research scientist. You have plenty of opportunities, of course. So these are all the agencies you can always look for. And if you have some special skills, like a scientific writer, or media sectors, if you are interested in taking some documentation both on the videography as well as for writing documentation, and there are many animal-based journals. So there are many journals where you have a lot of opportunities to employ yourself in those journals where they will be primarily utilized to you for writing articles for animal magazines, daily newspapers, as sometimes editorial assistants, and you can also do your own blogs for commercial websites, and you can also do some content creators for many conferences in biological sciences. Also, a kind of a TV programs would have listened to the Discovery Channel, National Geographic Channel, Animal Planet channels, without the background of the people who employed in those channels and the animal science background, it will be very difficult for them to survive. So these are all other some special skills if you provide, you can yourself create employment opportunities. And apart from that, there are again opportunities like you can serve as technicians in laboratories, many national laboratories, many biological laboratories, and many environmental science laboratories, many clinical laboratories, that is medical laboratories. You can employ yourself as technicians. And there are forensic science and toxicology departments, which primarily look for some special people to do a kind of research as well as for doing some kind of other related areas work. So you'll have a lot of chance. And there are some industries like food science and nutrition industries, and you have pharmaceutical industries. So these are all the areas one can always look for. And this is about the various avenues I told you on both the private as well as the public sector undertakings in India. And thirdly, what I say is for zoology students, they have a better opportunity than what I feel is than the physical sciences and chemical sciences, and they have several self-employment opportunities, especially in sericulture. Areas like sericulture, where you have a separate ministry in India. It is the Ministry of Commerce and Textiles. They look after, and there is a separate board which is established, National Sericulture Board, which is at Bangalore, and there are many research centers, and its headquarters is at Mysore, CSR and TI, and there are many other substations throughout the country. You can get yourself trained in those areas. You can also become an entrepreneur. If you have even 50 cents of an acre, that would be sufficient for you to make yourself self-employed through sericultural activities, where you can always do floriculture as well as silkworm rearing. And apiculture is another area which will, of course, in addition to the primary employment, this will give you a lot of other opportunities for you to earn more. And we are very well known about, we have a very large coastal regions, aquaculture, mariculture are again a lot of avenues. Of course, there are some avenues that are available with reference to lag culture in some of the restricted areas or regions of our country. Vermiculture is a very emerging area. We now talk about organic farming and all. Vermiculture is definitely going to be an important area in organic farming. And addition to that, poultry farming, in which we are all well versed nowadays. You have fish farming very recently. There are some fishes which are being grown, like aquarium fish maintenance. So these are all various other areas by which one can always be self-employed on their own. The zoology students, they have a lot of these kind of self-employment opportunities. So this is what I would like to, with this lecture series, I would like to highlight these areas, research as well as other avenues are more for zoology students than any other students. And I think the deliberations, what you are going to actually hear today with reference to primarily from Dr. Tyagarajan, I came to know that there are experts who are going to talk more on applied zoology, how we can get self-employed after their undergraduate or postgraduate programs. I think with these highlights or glimpses what I make on the employment opportunities or the scope for the zoology students, I congratulate the team at Vivekananda College as well as Dr. Tyagarajan for kindly giving me this opportunity to interact with you, and I wish you all the very best, and I am very happy to inaugurate this webinar lecture series. Thank you very much. Thank you.
Thank you, sir.
I think a good
Yes, yes.
start is half done. You have done our job more easy, sir, giving enthralling such and voluminous information on the scope of zoology. I think the apt person for to inaugurate this session will be we are very happy to be we are happy to have you in our inauguration, sir. Thank you so much, sir.
Thank you. Thank you. Thank you.
Thank
Can
you,
I leave now or?
Yes, sir. Yeah.
And I'll join after some time.
Yes, sir.
I'll
Yes, sir.
join
Thank you, sir.
Thank you,
I have
sir.
a work. I'll come.
Thank you,
Thank
sir.
you, Dharanja.
Thank you, sir.
Thank you. So now we are moving on to the invited speaker session. We have the first session by Abhijit Mitra. So Abhijit Mitra, sir, are you there?
Yes, yes.
Yes, sir. Okay, sir.
Okay.
I'm very happy to introduce Abhijit Mitra, sir. Dr. Abhijit Mitra, Associate Professor and former Head Department of Marine Science, University of Calcutta, India, has been active in the sphere of oceanography since 1985. He obtained his PhD as Net Qualified Scholar in 1994 after securing gold medal in MSc Marine Sciences from University of Calcutta. Since then, he has joined Calcutta Port Trust and WWF in various capacities to carry out research programs on environmental science, biodiversity conservation, climate change, and carbon sequestration. Presently, Dr. Mitra is serving Techno India University, West Bengal as the Director of Research. He has, to his credit, about 553 scientific publications in various national and international journals and 42 books of postgraduate standards. Dr. Mitra is presently the member of several committees like PACON, International, IUCN, SIOS, etc., and has successfully completed about 19 projects on biodiversity loss in fisheries sector, coastal pollution, alternative livelihood, climate change, and carbon sequestration. Dr. Mitra also visited as faculty member and invited speakers in several foreign universities of Singapore, Kenya, Oman, and USA. In 2008, Dr. Mitra was invited as a visiting fellow at University of Massachusetts at Dartmouth, USA, to deliver a series of lectures on climate change. Dr. Mitra also successfully guided 38 PhD students. Presently, his domain of expertise includes environmental science, mangrove ecology, sustainable aquaculture, alternative livelihood, climate change, and carbon sequestration. Thank you for the wonderful, thank you, sir. The biodata goes for a long list, and we have cut short with few. Here is a voluminous biodata of sir, and he is an apt person for this first session on polyculture and seaweed formulation. Over to you, sir.
Thank you. Am I audible?
Yes, sir.
Okay. Then you have to enable me to share the screen.
Yes, sir. You are now host, sir. Co-host.
Okay.
You can share your screen.
Okay.
Is it visible?
Yes, sir. Yes, sir.
Okay. So first of all, I would like to give my heartfelt thanks to the organizer for giving me this chance. Uh, I must say that I am so grateful on behalf of all the participants from the state of West Bengal. Vanakkam, and my greetings to all of you. The first thing is that I am so happy to see that from our neighboring country, some participants of the Joint Secretary rank and World Bank consultant have also joined from Bangladesh. I have seen Mr. Shama Prasad Bapari and Mr. Emin Sarkar, who are from the Bangladesh, our neighboring country. They are working extensively in Sundarbans, and I'm glad to see that they are also present over here to see our lecture, to see our, so that we can have an interactive session between our neighboring countries and between both the countries. Because we have mangroves both in India as well as in Bangladesh. In Bangladesh, we have the bigger chunk of mangroves of 62%, whereas in India, we have 38%. So I'm glad to get their views and to get their participation. Now, I have divided my entire lecture into two parts. First one is about the polyculture, and the second one is the innovation in the fish feed preparation. Now, if you see the statistics of the aquaculture production, it is, you see, the capture production and the aquaculture production. Capture production means you have got a limitation. If you see it with the orange graph, you will see that after that it has become stagnant, because after capturing and capturing and capturing, there will be overexploitation, and then the graph becomes zero. The slope becomes zero. But if you try to increase the protein content, you will see that the graph is increasing. Why you have to increase the protein content? Because today the planet Earth is sustaining 7 billion population, and in 2050 the prediction is there, it will exceed 9 billion population. Now, how to get this extra protein? Definitely it is from the fish protein, because it is one of the most digestible and most easily accessible. Now, if you see the nature is our best teacher. If you see the nature, there is not only single species. You see crabs are there. You see so many diverse species are there. So the nature promotes polyculture. Polyculture means, poly means many. And in nature, there are so many species. So polyculture is there in nature, and it is the teaching of the nature. Then, if you go to some mangrove spaces, you will see the color of the water is green in color. Why the water is green in color? Because of the presence of the phytopigments, which are the chlorophyll pigments from the phytoplankton, the base of the food chain. And on these phytoplankton, the zooplankton feed, and on the zooplankton, the fishes feed, and therefore this is a concrete food chain spun in the estuarine and coastal areas. India is having 4,871 kilometer total mangrove, out of which West Bengal Sundarban is having 2,120, about half of the mangroves. I think you have heard of West Bengal Sundarbans, and I will come in details on that. Now, apart from the fish species, you are also getting crabs. Crabs is also an important aquacultural species. Its cost is about 600 to 700 rupees per kg. The Scylla serrata, they are very important. And you get a lot of crabs along with other finfish and shellfish in the aquacultural practice. Now, what is polyculture? It is basically the culturing of more than one species of aquatic organism in the same pond. And it is based on the concept of utilization of different trophic levels. That means we don't want to lose any of the levels. We want a greater production to sustain these billions of population, this rising population. So that every niche has to be utilized properly. And that is the basic aim of polyculture. Now, there are certain merits of polyculture. maximum fish production is possible in intensive polyculture. What is intensive polyculture? Suppose you have per meter square, you can stock X per meter square. If you make it X square or X cube, then there will be the scarcity of oxygen. There will be the scarcity of natural food. So in that case, you have to go for aerator. You have to give oxygen through special mechanical method. You have to provide extra feed. And that is polyculture, to get the maximum production. That is intensive method. There are also semi-intensive method, but nowadays intensive method is used through various technologies we are adopting. And even the laser is adopted to kill the virus, to kill the bacteria and the spores. You are getting more profit from the fish farming. It creates many employment opportunities. And intensive polyculture is fully controlled by the farmer. More fish can be cultivated and produced from a very short place because you are utilizing every sectors, every trophic levels. Now there are certain demerits. The probability of the disease is very high. Sometimes it is not possible to get a big size fish. And there are also chances of disease in the polyculture system. Now, compatibility of species is very important. Which species are to be cultivated? I give you a very small example. See, today among the participant list, I was just going through this participant list. I found one of the participant, Navonita Pal. She is doing a research on mangrove seedlings. But the thing is that she is not compatible with other researchers. So when she goes to the field, there is certain sort of disturbance, there is certain sort of quarreling, there is certain sort of hitch and all these things. So in that case, I, being a guide, do not get the maximum production. I send the scholars with some motive, but if there is some disturbance, if there is no compatibility, I do not get the maximum production. In that case, if I send Abhirup Mitra or Prasenjit Pramanik, they are very compatible. Abhirup may not be a PhD scholar. His background is basically commerce and chartered accountancy. But with his passion, when he goes to the field, when he drives into the subject, when he goes into the aquarium manufacturing and other aquarium culture method, Prasenjit Pramanik, they are compatible. So compatibility and synchronization is the basic backbone of any production, of any business. That is very important. I'm just joking. I'm not telling that Navonita Pal is not always compatible. Sometimes she is also compatible. But the thing is that this is the basic backbone. If you culture, for example, you have heard, I don't know whether you have heard of vetki, that is a carnivorous fish. If you culture vetki with prawn, what happens? The vetki is being a carnivorous will attack all the prawns. And you know, in prawns, what happens? After a certain period of time, they go for the molting. Molting is just like the changing of the dress. When you become fat, you have to take a new shirt. You cannot use the old shirt every time. You have to use a new trouser. So the molting is the process. When the prawn goes, the exoskeleton, the chitinous exoskeleton, they start changing and a new exoskeleton is formed. But during this phase, the muscles of the prawn are very delicate and they are very, very lucrative item for this vetki and other fishes. So I have to see that who are very compatible, and therefore the compatibility of synchronization is the basic backbone. Now, first thing, what you have to do if you go for the polyculture, you have to control the harmful aquatic plants and animals because sometimes they are very toxic. Then you have to eradicate, you have to destroy the cannibalistic and unwanted fish. For example, I have told about the vetki. Then you have to put the lime to maintain the pH, because sometimes the pH goes down of the water about 6 to 7. You have to make it 8. Then you have to apply fertilizer, NPK. Why? Because this fertilizer actually promotes the bloom of the phytoplankton, and then the food chain starts. I have told you the phyto-based polyculture, phyto-based fish culture. So the phytoplankton has to be promoted, and this can be promoted by applying fertilizer. Then you have to go for the stocking. Stocking may be of two main types, semi-intensive variety and the intensive variety. But for the intensive variety, you require a very high-tech method. You have to use laser light, you have for destroying the bacteria, you have to use the aerators, you have to use the artificial feed, which are rich in antioxidants and promote the growth. So these are very important. So on this, if you have the availability of these things, then you go either for the semi-intensive stocking or for the intensive stocking. This is our case study zone. I have told the Sundarbans. Sundarban is an area which is having a mangrove area of 9630 square kilometer, 9,630 square kilometer at the apex of the Bay of Bengal in the lower Gangetic Delta, supporting 34 mangrove species. And if you see, this Sundarban is a very, very cyclone prone zone. On 20th May of this month, of this year, there was a severe cyclone known as Amphan. I don't know whether you have all heard of it, but all the fisheries were destroyed. Many fishes were killed. And today also West Bengal is suffering for a very economic loss. And this is a high cyclone prone zone because which originates from the Bay of Bengal. These mangroves act as the line of defense. And moreover, why I am telling of the mangroves? Because the mangroves litter, I mean the decomposed parts of the mangroves, when they fall on the estuaries, on the coastal waters, they degrade and breaks into nitrogen and phosphorus, which are the natural fertilizer, and they promote the phytoplankton growth. So mangroves are important in two ways. Firstly, they prevent the cyclonic depressions, they prevent the natural disasters, they prevent the vulnerability, and at the same time, they promote the fertilization of the aquatic phase and help in the growth of the fish production. This is Sundarbans, the only place in the entire planet where you get the mangroves and the tigers in a very, very compatible way. Panthera tigris tigris, the Royal Bengal tiger. Nowhere in the planet you get such type of compatibility, such type of combinations. And we had, after the Zoological Survey of India, they made a scat analysis. They found that the tiger also consumes the crab. So this is a unique ecosystem where you get about more than 300 species of fishes, and you get the tiger, you get the crabs, you get the mangroves. So these are the teachings of the nature. Everything has to be compatible. Everything has to be synchronized. This is the Sundarbans, and this is a map of the Sundarbans where you know there are 7 estuaries. All are coming and meeting the Bay of Bengal. So this is just a features. Now these are the compatible fish. The first one we call in local language Parche, which is scientifically known as Lisa partia, and the other is the freshwater prawn Macrobrachium rosenbergi. So these are very compatible species. Why they are compatible? Because the Lisa partia, they often consume from the column the various zooplankton and the phytoplankton. And this Macrobrachium rosenbergi, they are the bottom feeders. So the excreta of this lizard, they are consumed by them. Otherwise, if they would not have been consumed, then the excreta, which is rich in ammonia, they could make the water toxic and cause several types of diseases. So this is the compatibility. You have to know the biology. We have to know the composition of the excretory product. You have to know the food cycle of each species to start the business in the fishery sector. You see, I will come with this. This is the picture of a prawn fed with Enteromorpha intestinalis, which is a seaweed. Now, when I'm speaking of the applied zoology, I'm not restricted to zoology only. Today, none of the subjects have the barrier. All the subjects have become interdisciplinary. The barriers have dissolved. As for example, I have told Avirup Mitra, who is sitting over there, he is by profession going for a chartered accountancy course, but he maintains his own aquarium shop. Very beautiful collections he's having, and he's having in the heart of the city of Calcutta. He maintains big shops over there. So it's by passion. It's not restriction. It is not, there is no restriction, and it's not restricted to any subject that I have to study zoology. If you have the passion, you can grow fish. It does not mean that you have to become a zoologist. So here is an interdisciplinary approach where the botanist can also fit very nicely, because the food source is the enteromorpha, which is a seaweed. Now, we have made a culture over there, prawn and the lizard, what I have told. And we have found that after the cost benefit ratio, because after the end of the day, you have to see that what money you are getting per hectare, you are getting 1.57, 1.58 if you go for the monoculture. But if you go for the polyculture, your income is increasing. And also there are certain biological indicators I will teach you, but in a very slow way. For example, if your fish is good, how you can see, how you can say that your fish is having a better growth? You know, you take the weight of the fish. You take the weight of the fish. I have shown you that this is the pan balance. This is the balance. It costs very less, about 800 to 1,000 rupees. And you take the weight, you take the length. Then you divide the weight with the cube of the length. It is the ratio of weight with the cube of the length. If it becomes nearer to 3, that means it is having a very good growth. You see, if you see the value, we have made an equation. Here also you see the mathematicians are coming into the forefront. This equation is having 2.95 and this is having 3.01. This is weight and length. We have transferred it into logarithm, an exponential logarithm model. And you see that the growth is almost isometric in case of prawn and also in case of lizard. That means the compatibility is there. If the compatibility would not have been there, it would go down below 2.9 or 3 and could have touched about 1 or 2 like that. That means your growth is not uniform. So you see, this is an interdisciplinary approach where the mathematics is coming, where the botanists are coming, where the zoologists are coming, and even you have to look upon the environment, the environmental water quality. So the environmental scientists are also coming in the scenario. So there are chances of employment from all sectors in this livelihood programs. Now I am coming to the next phase of my lecture. The first phase is over, the merit and scope of polyculture, because I'm very systematic, so I'm doing this. Innovation in fish feed preparation. What are the innovation? How you are going to make the fish feed? If you want to make the fish feed, what you want, if you want to make the fish feed, then you want, you know, a rice bran, then the dried fish, then the wheat bran, then something chelating agent. All these you mix up and put it in the pelletizer. It comes in the form of a pellet with a protein of 25 to 26% for the floating fish and something more for the sinking types of feed. So this is the fish feed preparation method. But what happens, you know, when you are mixing the dried fish as an ingredient of the fish feed, sometimes it happens the fishes are not taking the feed at all due to some reasons or the other. They are not getting that appetite, so the fish's feed remains in the aquarium base or at the pond bottom. And what happens in that case? The ammonia generates, the water becomes toxic. We say that diseases are happening, but the diseases are happening because of the poor quality of the water. In that case, if you replace the dried fish ingredients with the enteromorpha, with the plant-based protein, it is better. For example, soybean, it is having high protein. If you put the soybean much more instead of the dried fish dust, then it is good for the environment. You see, this is a very old times. We have started this type of factory as an entrepreneur about 20 years back in a place called Canning, where we started this factory. At that time, the instruments were not sophisticated like this. And this is the Enteromorpha intestinalis. Plenty of Enteromorpha intestinalis are available. Even in the aquaculture farm, they are as the waste products. They have got no value. The farmers do not know the idea that what huge amount of protein this seaweed is having. this seaweed is having about 13 to 17% protein. And therefore, we made a plan to dry this seaweed. Firstly, we sterilized it, dried it, and then we put it as a feed ingredients in the fish feed, and we then tried. You see, this is the seaweed reservoir in the West Bengal. Of course, if you go to a place called the Digha, you will see that lots of these enteromorpha are found in the nature. With the gastropods, they are thriving over here. The mollusks are also there. So you see, in the nature also, they are all in a compatible mode. There are botany, there are zoology. This is the embankment, and you will see this is some engineering construction over there. So what I'm trying to tell is that the subject has become multidisciplinary and interdisciplinary, and with a holistic approach, you have to approach this fishery sector. You see, our scholars put ropes in a coastal area, and after 3 months, you see these ropes are carpeted and covered with huge amount of enteromorpha. So there is no scarcity of the raw materials. We can go for the fish feed preparation, and this is another innovative employment opportunity. We can go for the fish feed preparation with this dried enteromorpha species, with this dry seaweed. This is usually, these are the ingredients: rice bran, wheat bran, fish meal, shrimp meal, then sunflower, mustard oil, cake, garlic paste, then enteromorpha, binders, and the shark oil. But if you replace this fish meal and shrimp meal, why? Because I have told that they cause some toxicity in the aquatic phase. And if you just replace it with the seaweed dust, then it gives a very good results. The environment is upgraded. That is the main thing. The water does not become toxic. The water does not become poisonous. The water becomes healthy, and then only you have an isometric growth with a value of W by L cube, W proportionate to cube of the length, with a value of 3 or nearer to 3. Now, we have made a histological structure. The students of zoology know that with a microtome section, we have made the analysis of the muscles. We have seen that, you see, whenever we have used enteromorpha, the growth has become very good. You see the muscle fiber, this bluish, these whitish things, in comparison where we have not used, you see the muscle fiber, they are very stiff and not very expanded, but you see it has become expanded. So this proves that plant-based proteins are more effective, not only to upgrade the water, but also in promoting the growth of the fish cultured species. So we have made also, but if you start taking the enteromorpha from the nature, it will not be a sustainable thing. There will be an over-exploitation. So we have made some enteromorpha culture in some places in Sundarbans, where you have stocked the brackish water and put the thallus, and then they started growing luxuriantly. Now, there is another important thing. Today, a very important issue is the global warming. You know, in the globe, previously there was a time when the atmospheric carbon dioxide was about 300 ppm, but now it has exceeded 410 ppm. If you see the internet now in the Google itself, you will see today it has crossed 415 ppm, 415 ppm. The permissible level is 350 ppm, 350. So this is global warming, which is causing all the imbalances on the planet Earth. The sea level is rising, the acidification is occurring, there is weather disruptions, suddenly the rain is coming, suddenly the rain is going, there is flood in Mumbai, there is flood in Assam. You see, there is huge high temperature which is not expected. So these are the weather abnormalities. All these are because of the climate change. And the culprit are the greenhouse gases, the excess carbon dioxide in the atmosphere. Now, why I am telling all these things? Because if you culture seaweed, this is a figure that how the carbon dioxide have increased over a period of time from 1984 to 2016. You see it has exceeded 400 ppm in Sundarbans only. In all the seasons, whether it is pre-monsoon, monsoon, or post-monsoon. So this carbon dioxide is absorbed if you culture the seaweed, because seaweed can absorb the carbon dioxide, because, you know, these seaweeds, they photosynthesize, and for photosynthesis, you know, the basic raw materials are the carbon dioxide, sunlight, and water. And this carbon dioxide is fixed in the seaweed with the help of an enzyme known as rubisco. So the culture of the seaweed is not only helping to upgrade the aquatic phase, it is also promoting the growth of the cultured fishes. At the same time, it is also upgrading the environment and reducing the greenhouse gases. With these few words, I am thanking to all of you. I am thanking to Ramakrishna Vivekananda College of Mylapore, Chennai, for giving me this opportunity. Thank you, all of you. Thank you.
Thank you, sir. Thank you so much. Abhijit sir, shall we take some questions?
Yes, definitely. Definitely.
So participants who are wishing to have a question can raise their hand. Any questions, clarifications? Any questions, clarifications?
Yeah.
Any questions from the participant side?
So I think there is no question.
Yeah, it was very much clear, it seems, sir. So, Tyagarajan sir,
Ah,
I think you can give the concluding remarks.
Thank you, Dr. Abhijit sir, for your wonderful presentation. It was really a beautiful presentation on the Sundarban mangrove ecosystem with a lot of nice photographs of animal. In fact, one particular picture that caught my interest was the prawn, because that was my PhD model also.
Okay.
So it was really nice, sir. Sir, your first talk on mangrove ecosystem was very informative, wherein you discuss about the scope of polyculture system as well as animals that are used in polyculture. Your second talk on feed formulation, their composition and the economic importance. And what was really interesting was the role of seaweed in coastal ecosystem regeneration and conservation was very novel, and it was really an eye opener for all of us. I am sure that not only the students of zoology, but students from other disciplines will also be interested in becoming entrepreneurs in aquaculture as well as polyculture ecosystem.
Yes.
I once again thank you, sir, for your wonderful presentation. Thank you, sir.
Thank you. Thank you very much.
Thank you, sir.
Okay. Thank you.
Sir, there is a question.
Okay.
Let me ask. What is a MSc student's opportunity in this field?
Firstly, I would like to tell that they can be a self-entrepreneurs, number one. Number two, MSc in which field? Who is asking the question, MSc in which field?
Generally, sir, an MSc student.
MSc in the biological field, for example, in zoology,
Maybe.
botany. So if they are interested, there is an CSIR institute, CSMCRI, Center for Salt Research Institute at Vabnagar, Gujarat. They are doing wonderful research on the seaweeds, and also they have got a very good field station at Tuticorin, where the lots of seaweeds are cultured and they are used for various other purposes, not only for the fish culture, but seaweeds are used for making various food items and other things. So there are a lot of industrial applications. Even I don't have the presentations today. Even seaweeds are now used for making shampoo, shaving cream. So there are also opportunities to being absorbed in the pharmaceutical industries also, because many pharmaceutical companies are using seaweeds for making the shaving cream, for making soap, for making shampoo, because they are having iodine and good antioxidants. So there are a lot of scopes in various things, and also in the pollution control board, because seaweeds and other biological entities serves as the indicated species. They can indicate the health of the environment. Particularly when we are consuming the fish, it should be monitored and checked that how much better and good for the health. What is the level of accumulation? What are the organs, the gills, the muscles, the bones inside, which are more accumulating more in comparison to other? So we must discard that. And all these are taken care by the pollution control boards. So they have set the permissible level, and therefore there is chance to get involved not only in the research, but also in government sectors like pollution control board, state and central pollution control board, and also in various other agencies like fisheries, forest department, etc. Thank you very much.
Sir, any other examples of polyculture?
Yes. If you put carp, means rohu, katla, and also the prawns, it's a very good example, because rohu, katla, one is the surface feeder, other is the column feeder, another is the prawn is the bottom feeder. So it's a very good example where all the trophic levels are used. Here is one participant, Dr. Pritam Mukherjee and Dr. Prasenjit Pramanik. They made an excellent study in West Bengal, and they have created a model of this of a nice compatibility of these three species: rohu, labio rohita, carp, katla, and Macrobrachium rosenbergi. This is a very good example of polyculture in the freshwater system. Thank you.
So what is the overall cost of setup?
The overall cost of setup depends on the dimension, but per hectare, the overall cost varies from 35,000 per hectare to about 1 lakh, depending on the semi-intensive and intensive variety. And you get the return within 6 to 8 months, almost double.
Sir, what are the possibilities of ornamental fish aquaculture?
Ornamental fish aquaculture has the possibilities, but now because of the COVID lockdown phase, it has come very down. And Abhirup Mitra, who I'm telling about, he is a specialist in that. He brings ornamental fish from South Asian countries, breed them, and also sell them. So if you just Abhirup, if you can unmute, I will request the organizer to unmute Abhirup Mitra so that he can speak few words that what are the possibilities, because he is doing a hands-on training on that.
Abhirup, sir. Abhirup Mitra.
Abhirup Mitra.
Yeah. He can unmute himself, sir.
Okay. Abhirup, you unmute.
So am I audible?
Yeah.
So basically what I do is mainly I deal with exotic fishes, but recently I have started an act for doing with native fishes. So the surprising fact is that all over the world there is around 36,000 types of fishes. And the surprising fact which I have found out, that corresponding to an exotic fish, we have the same variety in India. Like suppose we have a killifish which is available in the Amazon River in Brazil, the same killifish is available in the Western Ghats, and we call that lampai fish. But these Indian fishes were never brought to the world. But now recently the demand for that Indian native fishes to be kept in aquarium has gone up by a huge amount. And the main Indian market which runs is with the chana species, which is known as the snakeheads. So in Bengal, we have the Bengal snakehead, which is known as the chana gachua. It's a dwarf snakehead. And recently another snakehead which was discovered in West Bengal, Jalpaiguri, in the year 2018, is Chana brunei, or what we call it as chocolate snakehead. So I have kept a pair in my room. So I have been researching on that. Hopefully it will breed in another month. So if it's breed, then it will be an achievement for me. And the snakeheads are snakeheads, and we have a lot of barbs in India too. The Western Ghats, it's full of fishes. So not only for the eating purpose we can supply fishes, but for ornamental purpose as well as keeping purpose, like a hobby purpose, Indian, we can also be a competition to the world.
Oh,
Okay.
thank you,
Thank
Abhinav.
you.
So
Thank you, sir.
what I was telling is that, so you see, his background is chartered accountancy, but you
Okay.
see his passion, and he knows a lot of about the other fishes, the scientific names and others. Some things that the zoologists should be jealous of him. So it's about the passion, and he is an entrepreneur in West
Oh.
Bengal. He is the entrepreneur at this age. So this is something wonderful. So there are a lot of opportunities in the ornamental fish sector also.
Okay, sir.
Okay,
Thank you, sir.
thank
Thank
you.
you so much. So
Thank
with
you.
this, we have come to the end of this session. And
Okay.
thank you so much for your wonderful presentation and interaction, sir. Thank you so
Okay.
much.
Okay. May I now have the opportunity to leave? Will you just
Yes, sir.
permit
Thank you,
me?
sir. Thank you, sir.
Thank you.
Thank you, sir.
Thank you. Thank you very much. Thank you.
So, dear participants, as I have informed in the Telegram group and the WhatsApp group, so now we will have a live quiz now based on the information or the presentation given by Dr. Abhijit Mitra sir, and I'll share my screen with the code for the game, and I request everyone to join in that. Hope all the participants have registered yourself into that mail, into that quizzes.com. So this is the enter code, and you have to enter this code into your join game option. So in that you have to enter this code 287606, and I have one participant, Abhirup Mitra, then 2, 3. I want everyone to play this game because this will be taken into for your attendance. So we have around 239 participants, 38 participants. So I want everyone to actively participate in this. Come on, participants. We have 237 participants, and we have only 109 so far. So only 156 have joined. What about the rest? So we'll wait for a few more minutes.
சிவச்சந்திரன் சார்.
சார் சொல்லுங்க சார்.
சில பேர் லிங்க் கேக்குறாங்க, உங்களுக்கு கிடைக்கல.
எந்த லிங்க் சார்?
இந்த குவிஸுக்கு
ஒண்ணும் இல்ல சார். சார் for the participants you have to separately log in to joinmyquiz.com or quizzes.com, join game with your registered email ID. Then you have to give your date of birth, then you have to join game, then enter this code. So it will take few steps. I have already mentioned, or I have already given these steps in the group also. So it will take some time, but try to register and join the quiz. joinmyquiz.com or quizzes.com. Either of the websites, that is either of the topic you can put in this is what to say in another separate browser. 190. What about the other 40? So once it touches 200, we'll be starting the game. That's good. We have 202. So participants, this is just an interaction away, that is interacting with the participants. It's a form of quiz. We'll be having only 5 questions based on the talk which was given by the invited speaker. So you need not worry about what is going to be or something which is going to be tough. It will be very basic questions from what the invited speaker has given in his presentation. So if you have observed the presentation, then it will be more easy to answer it. So we have 209, 210. Satyagran sir.
சொல்லுங்க
சார். அதான் இப்போ இதுல வந்து யூடியூப்லயும் ஒரு 100 இருக்காங்க. ஸோ இது 240யோட ஸ்டாப் பண்ணிக்கலாமா இல்ல வெயிட் பண்ணுவோமா?
இன்னும் என்டர் ஆயிட்டே இருக்காங்கல்ல.
அதான் ஆயிட்டே இருக்காங்க.
224 வந்துருச்சு.
அதான்
இன்னும் few minutes வெயிட் பண்ணலாம் சார்.
ஓகே சார். So we'll start by 3:45.
Yeah.
2 minutes more. The participants will be starting the quiz by 3:45. So try to enroll yourself. We have few participants in YouTube also. I think it's done. So we are starting the game.
முடிச்சிட்டாங்களா சார்?
இன்னும் 322 பிளேயர் சார் விளையாடிட்டு இருக்காங்க.
Oh,
269 ஓவர்.
ஓகே.
ரிமைனிங் ஒரு 40 இருக்கு. So with this, we'll be ending this game. So here are the winners. So first, Deba Smita are live game second, and third Harish. The others have secured 100%, but the timing was too long, so they are behind the first three participants. So thank you all for participating, and we'll be having another quiz at the end of second session. Thank you. Thank you for all the participants who have participated, and congratulations to the top three winners. Thank you. Now we move on to the second session by Dr. B. Gunalan. I invite Dr. K. Rajagopal, Assistant Professor of Botany, Ramakrishna Mission Vivekananda College, to introduce the speaker for the second session.
Thank you, Tyagarajan, and thank you, Sivachandran. I'm very happy and privileged to introduce Dr. B. Gunalan, Assistant Professor. Currently, he is working as a Assistant Professor, PG and Research Department of Zoology, Kolunjiappar Government Arts College, Virudhachalam, Kadalur District. Actually, he was under deputation from Center for Advanced Studies in Marine Biology, Faculty of Marine Sciences, Annamalai University, Parangipetai. Professor B. Gunalan is a very good teacher as well as researcher. His field of specialization include aquaculture, crustacean diversity, and conservation. Apart from teaching, he is actively involved in research, which is reflected in his biodata. So far, he has produced 5 PhDs, and he has published 41 research publications in national and international journals. Totally, his research articles cited 313 times with H index of 12 and I10 index of 13. Apart from research publications, he has published 2 books. He visited several countries, to mention a few: Sri Lanka, Vietnam, Thailand, and few more countries. Before he entered into teaching line, he served as a chief scientist in Sagar Sambada Fisheries Research Vessel, Government of India, 290 cruises. Apart from teaching and research, he is a very good human being. I had the opportunity to meet him in Pondicherry University. When the Department of Zoology approached me for a person who can give and motivate the young students, I thought of Professor Gunalan. I welcome you, Professor Gunalan, for this webinar.
Thank you, sir.
I want to thank Dr. Tyagarajan and his team, particularly Dr. Sivachandran, for the opportunity given to me to introduce such a person. B. Gunalan, now over to you.
Thank you, sir. Thank you for your nice words.
Sir, you can share your screen, sir.
Yes, sir.
Have I given you the host?
Sir, you have to disable, sir. You have to.
Yeah, 1 minute, sir. 1 minute, sir. I'll make you co-host, sir. Now you can share the screen, sir. Yeah.
So is it visible, sir? My screen is visible?
Yes, sir. Yes, sir. Yes, sir.
Yeah, good evening, one and all. First of all, I would like to thank the principal sir, Dr. K. Sedu Sankar sir, RKMVC'S College, the co-convener of the program, Dr. Tyagarajan Raman sir, Assistant Professor, Head of the Department of Zoology, and also Organization Secretary, Dr. S. Sivachandran sir. He is a person keep on talking with me past 10 days. I would like to thank my good friend Dr. K. Rajagopal from Botany Department. Thank you all for giving me the nice opportunity. I would like to present the sim culture techniques and its scope. I want to, when I got the topic, just I started asking myself, why this webinar? Why we have to know about the stream culture? Because we can control the over exploitation or conserve the wild stock. No need to depend on the capture fishery. We can control the nutrient deficiency, because today, as an FAO report 2020, more than 2 billion people are under micronutrient problem. So, and also this particular sector giving employment opportunity to many people. And also our Prime Minister, Ji, talking about a blue economy. What is blue economy? Because the sustainable development only who can give, only agriculture and the aquaculture department, aquaculture sector only can give the sustainable growth and the economy to the India. Okay. So I want to tell before going to the section, I want to tell the history of the shrimp culture. The shrimp culture started by the wild capturing from the estuary river. They have captured the small tiny shrimp. They have kept in the paddy field in the year. Scientific way of culture started in 1930 with Pinus japonicus. Now it's called Marsupinis japonicus. In India, the culture was traditionally practiced in West Bengal and Kerala previously. So West Bengal, they used to culture the shrimps, and also in Kerala they used to culture the shrimps. in the paddy field. That's the West Bengal used to call Biharis and Kerala used to call it Pokali. So the commercial aspect of shrimp, Central Institute of Marine Fisheries Research Institute in 1970, they have started the culture commercial aspect. So followed by Marine Product Export Development Authority in 1988, with the help of American technologists, they have started the culture in Orissa and Andhra Pradesh in scientific manner. In the year 1990 to 94, the shrimp production was drastically increased. It means 40,000 metric ton to 1,43,000 metric tons. The major shrimp was cultures black tiger prawn, they call Pinus monodon. So due to disease outbreak in the white spot syndrome virus in the year 1995, after 1995, the production was drastically decreased because of WSSV, white spot syndrome virus, as well as anti-dumping crisis. What is anti-dumping crisis? Anti-dumping crisis is nothing but the farmers started using lot of antibiotics because when we were exporting the shrimps, the European countries as well as American companies, Japanese, they used to check the antibiotic residue in the animal body, shrimp body. So they have the limitation. So it's class, when it's class limitation, they started putting the charges. That means they won't reject the container, but they will put the duty, high duty. We cannot sell shrimps in good rate. In India, trial basis in 2003, white leg shrimp, Lithopenes vanami introduced. It's otherwise called Pinus vanami introduced because why they have given the trial basis? Because it's the exotic species. It's native of Latin American country. From 2019 onwards, officially allowed to culture with proper biosecurity. When I speak to the black tiger shrimp, that time they're not talking about biosecurity. But when they started allowing Lithopenes vanami to culture, they're talking about biosecurity because Lithopenes vanami is an exotic species. When we are culturing, when we are transferring the exotic species from other countries, what will happen? It might carry some of the viral diseases because still as on date for WSSV, white spot syndrome virus, there is no medicine like our COVID-19. So we are struggling with that corona. So we don't have any medicine. So like that, this from 1995 onwards, as on date for white spot syndrome virus, there is no medicine. Once the shrimp got affected by white spot syndrome virus, we have to harvest the shrimp because otherwise all the shrimp will get die. Presently, India is producing 8 lakh metric tons in culture shrimp. So it's, if you calculate 3.3 tons per hectare per year. So and also now for the this particular shrimp culture industry growth is very aggressive because the support of so many feed companies in Indian Council of Agricultural Research, Marine Product Export Development Authority, Rajiv Gandhi Center for Aquaculture, Coastal Aquaculture Authority, National Fisheries Development Board, and Department of Animal Husbandry and the state fisheries and state fisheries. So coming to the topic, why I have chosen that particular topic? Because this particular shrimp farming or shrimp culture is an important economic activity in many countries, and also it's offer number of opportunities to contribute and also alleviate the poverty and also giving employment opportunity, and also it's controlling the over exploitation and also giving the food security to the country. So you can see the map. You see Saudi Arabia, India, Myanmar, Thailand, Vietnam, South Africa, Madagascar, then Singapore, Sri Lanka, Malaysia, Philippines, Australia, Taiwan. Almost maximum of the countries they are culturing the shrimps. Okay, why we are particular about shrimp? Because if you see the global trend of animal meat eating or animal meat production, if you see beef or pig meat or poultry meat or sheep meat, automatically there is a saturation point. But in the case of fish or shrimps or any crustaceans, so you can see the growth pattern. It's in orange color, is yellowish color. It's increasing trend. So that's why the shrimp industry is growing aggressively. So if you come to the Indian scenario, so now India is the #1 shrimp producer in the world. So per year 8 lakh tons per shrimp production. That the value is 38,000 crores rupee value. That seed production in India 75 billion and feed production 1.8 billion tons. So come to the point, what are the key factors for successful culture of shrimps? How to make the successful shrimp culture? So there is here 4 key factors. One is seed, water quality, feed, and management. So if you come to the technical part, so pond preparation. So pond preparation, there is four methodologies there. So process involved drying the pond, scraping the pond, ploughing the pond, and liming the pond. So if you see, so this is the drying the pond. We have to allow the pond to dry in the sunlight because it will help to eradicate all the unwanted microorganisms and also it will make the soil from the wet to dry. So scrapping, so there is 2 kind of scrapping people used to do, manual and using the machines. So if you see, this is a manual scrapping. So using the labours and scrapping the soil 2 to 3 inches where the accumulated organic load waste is there we can scrap the soil. So remaining place, we can scrap the top soil and we have to remove from the pond. See, after scrapping, you can see the screen, lot of cake-like soil, cake-like structure. Because this one is, if you are not remove the scrapping properly, it will be lying in the pond bottom. If he is lying in the pond bottom, once he started pumping the water, what will happen? Automatically, this soil particle will dissolve with the water, it will create the problem. So scrapping by machinery, by using the tractor, with the trouser, we can scrap the soil. So ploughing the pond, why we have to plough the pond? Ploughing means we have to plough horizontal and vertical, we have to plough the pond. So ploughing the pond using the old traditional method also, and also we have another question, without dry, maybe some areas it on the pond bottom won't get dry, or due to the climatical factor, due to rain factor, we cannot able to make dry the pond. Such a case, how to prepare how to plough the pond? So what we have to do? So we have to use the cage wheel of tractor, and we can plough the pond. We can fill with 1 feet or half a feet of water. We can disturb the pond bottom, and after that we can pump the water out, or we can open the sluice and drain the pond. If you do like this 2 or 3 times, automatically the culture pond, which one you are going to put the shrimps, that particular pond, all the microbial load will be getting down. So why we have to plough the pond? So we have to plough the pond horizontally and vertically with the depth of 20 centimeter to remove the unwanted gases and also oxygenate the bottom soil and also make the discoloration of the black soil, color of the soil. So remove, and also odor, hydrogen sulfide water we have to remove. So and also increase the fertility of the soil. So there are 4 types of land they are using in aquaculture, especially in shrimp farm. That's one is calcium carbonate, calcium magnesium carbonate, calcium oxide, and calcium hydroxide. Calcium carbonate they used to call is agri-lime. Local name is agri-lime. Calcium magnesium carbonate is dolomite. Calcium oxide is stone lime. Calcium hydroxide is shell lime. So why we have to apply or use the lime? Because the lime is playing the major role, because it's increasing the soil fertility in such a way, because soil have the pH concentration, hydrogen and concentration will be vary depend upon the soil. Because some of the soil they have maybe the pH is more than 7 or less than 7. In the case of less than 7, we have to use calcium hydroxide or calcium oxide. In the case of the pH is 7.5 or 7.8 or 8, we can use the agri-lime or dolomite to fertile the soil. Okay, so when you are applying the soil, lime to the soil, what we have to do, you have to apply the lime when there is less wind action, that time we have to apply. Otherwise, the powder, the lime powder won't be fall into the soil. After application of the soil, what we have to do, lime, we have to one more time we have to plow the pond. Then automatically the lime will be mixing with the soil will get fertile. So when the word I'm using the biosecurity, because the biosecurity playing the major role, because when the exotic species Lithopenes venami allowed to culture in India, the Coastal Aquaculture Authority, they are very keen in biosecurity. The farmers used to get the license as well as they used to complete their ETP system as well as bat fencing and crab fencing. Because bat fencing and crab fencing, bat fencing is it's like a controlling the vertical contamination. Bat fencing and crab fencing controlling the vertical contamination. Oh, what about the horizontal contamination? The horizontal contamination, it will come through the man as well as from the bull star, from the mother prawn. Okay, so through that we have to become so conscious about the bat fencing, crab fencing, because the birds or crabs, the crab enter into the pond because the virus can live in the crab, because white spot syndrome virus, that particular virus, it is a crab is a host. So when it's entered into the water, automatically water will get infected. So what we have to do, we have to be very keen about the crab fencing as well as the bat fencing. So if you tie the net nylon rope in the pond, what will happen? The bats cannot fall into the water or cannot pick up any animal, or it cannot disturb the water column. So this is the bats carrying. So for this thing, we can use some old video tabs or a tape recorder, cassette tabs, or any polythene pieces. If you tie in the rope, what will happen? So it will create the sound. Through the sound, the animal won't disturb. The animal will get disturbed. It won't come. So this is the crab fencing. So we have to cover. From the bund, we have to 2 feet. We have to cover with the net. So no animal can enter into the culture system, including the sluice. We have to cover. So after completion of biosecurity method, so we are moving to pumping section. So the pumping section involved 3 important steps. One is filter bag, another one is pumping method and lab. So if you see filter bags, so you can able to see the filter bags, because why we have to have a long filter bag, as well if you are small, due to the water pressure and also air, that the water contain a lot of planktons. So what will happen? Automatically the holes will be get clacked and the bag will be get wrapped. So what we have to do? We have to clean the clean the bag simultaneously, as well as we have to make the bag also lengthy. So through that we can able to avoid unwanted organisms entry inside the pond. Okay, so you can see these are the unwanted organisms. If you are not properly filtered, so crab larvae or fish larvae or some tadpole larvae will enter into your system, culture system. Okay, why we have to pump one by one? Because we have to fill the pond one by one. Otherwise, what will happen? If you have 6 ponds or 7 ponds or 2 or 3 ponds, so we have to fill the pond one by one, minimum 60 to 70 centimeter water first time. So if you simultaneously, if you are opening all the pond at the same, what will happen? So all the pond will get filled slowly. So 1 centimeter, 2 centimeter, like that. So what will happen? Automatically through the sunlight will pass the water column. Through the water column, it will touch the pond bottom. So what will so automatically it will algal growth will be there. So we have to, for avoiding this thing. So because once we are preparing the pond bottom so clearly, if you are not careful about the filtration or filling system properly, so the time you have spent, the money you have spent in the pond preparation is waste. Okay, so these are the problem will happen. So you can see the picture where you see the algal growth. Lab means the algae is floating. You can see, you see like this. So water culture. So water culture is one of the important section because we are going to introduce the shrimps into the water column only. So for that we have to fertile the water. So how to fertile the water? So we have to through the line because water have the pH. The ideal pH for culture is 7.5 to 8 pH. So we have to know when the water is pumped water, there is no color. No color won't be there. So when there is no color, we have to create the phytoplankton production. That means phytoplankton bloom. If we are not creating the why, we have to create the phytoplankton bloom because when we are living in the house, we need a roof to cover. If without roof, you cannot sit because the sunlight will fall directly to the house. So like that, so we need a roof-like structure. So the shrimps will be happy when the phytoplankton bloom is good. So the shrimps will be very happy. So for that we have to use dolomite or agriland. Mainly we are using for increasing the phytoplankton production. We are using organic fertilizer as well as the inorganic fertilizer because we are using the inorganic fertilizer like urea, DAP, super phosphate. We have there is the ratio is there. We have to mix with the water. Then we have to filter the water. Then we have to apply entire the pond. The spreading must be the entire the pond. We have to apply properly. Then only the phytoplankton blooming will be very good. So organic fertilizer. What is organic fertilizer? Organic is nothing but the cow dung, rice bran, groundnut cake. With the help of yeast, we have to ferment with water. So overnight, the next day we have to filter the water. Then we have to apply into the bottom. It will help to develop the zooplankton because the animals, the younger animals, juveniles, they like zooplankton. So probiotic will come to later. So these are the inorganic fertilizer like urea, DAP, and super phosphate, rice bran, groundnut cake, and cow dung and yeast. So what is probiotic? Probiotic is nothing but is a beneficial friendly bacteria, which one is control the bad bacteria, and also it will give the proper digestive and digest the organic matter, water as well as the pond bottom. Okay, so the probiotic there is three type of probiotic is there. One is water probiotic, another one is for the pond bottom soil probiotic is there, another is for the mixing with the feed. The water probiotic, what it will do, it will maintain the yellow colony and green colony in the water column because yellow colony is beneficial bacteria, green colony is the harmful bacteria. If you see for what we have to apply soil with that probiotic because soil is if you are not properly because we are feeding animal waste is there, feed waste there, it will create the excess ammonia, nitrogen will be created. So what will happen automatically the bottom will be get polluted. So for controlling that particular pond bottom, we have to use probiotic which the species rhodopacter and rhodococcus will be very good to clean the pond bottom. Okay, so in the case of shrimp body, so mixing with the feed, we have to use some baseless species. So if we give, automatically the shrimp gut will be good, and also gut won't get any unwanted microflora infection. So these are the some product available in the market like pH fixer, multivitamin, and superbiotic baseless species. So these are the okay. These are the general common measurements given the parameters is maintained in the pond. So you see disaloxin 3.5 to 4 ppm parts per million. Salinity is 10 to 25 ppt is optimum salinity. Some places 15 to 25 ppt also optimum ppt. Water temperature is 26-32 degrees Celsius. pH is 7.2 to 8.5. Total nitrogen is 1 ppm. Total ammonia not less than 1 ppm. BOD is 10 ppm and COD is 70 ppm. Transparency is 35 ppm. Transparency is an indication of phytoplankton production. If the transparency is increasing, the phytoplankton production is less. If transparency is less, the plankton bloom is good. Okay, carbon dioxide and sulfates and alkalinity. So these are the optimal levels maintained in the shrimp ponds. So if the water color is good, automatically we will have the better growth rate because of the natural food. as well as the better survival rate, healthy, swim will be very healthy, the environment will be very good, and also uniform in size. Animal is uniform in size. So we have to check the zooplankton, how the zooplankton is there. So like that we can check. If you check like this, you can see that the zooplankton population will be more. I think some of them is scribbling the pad. It's disturbing. So seed stocking.
Excuse me, sir. Participants kindly avoid these type of things.
Okay, seed stocking. That particular seed stocking, it's important section because selection of seed is important, because already I told in the beginning itself 4 key factors: seed, feed, management, and management system. So through that the selection of good seed is important, because you are pond preparing properly, water culture is done very nicely, probiotic application done very nicely, biosecurity done very nicely. But if you are not selecting good seed, you cannot grow you cannot success the culture. So selection of good seed is important. The stocking method is important, and also we have to provide sufficient aerator based on the stocking density. So if this good quality seed will lead to good growth, then it will reduce the cost of production. So maybe per kilo shrimp for producing shrimp, 1 kilo shrimp for the 30 grams, hardly nowadays it's more than 120 to 150 rupees. Okay, so we have to be careful about the production cost. So physical observation, while you are checking the seeds, observation the hatchery tank. The hatchery tank, when you are visiting the hatchery will show the tank. See, we have the seed. So they will small bowl, they will show the seeds. So we have to check by eye visual how is the size variation of the seeds, and also swimming behavior of the seed, antenna and uropod, the tails is spreaded or not spreaded, how about the average length, any dead fish is there, like that we have to check in the system. So this, you see now that the seeds, like the seeds will be appear like this. You can see this post larvae is 15. You can see the spreading of uropod. So like that it must be spreaded. So the microscopic observation, this will be done in the laboratory, PCR laboratory. So they will check the gut ratio, then muscle gut ratio, then muscles, then any necrosis is there, any biting is there, any fouling organism is there, any internal parasite is there, and then they will check the seeds. So after checking the seed, everything is okay. So we can pack the seed from the hatchery. So depend upon the distance, maybe 5 hour traveling distance or 10 hours traveling distance. Based on that, we have to put the seeds, 1,500 seeds or 2,000 seeds. So when you are traveling long distance, we have to request the hatchery to reduce the temperature of the water, which one they are packing water. We have to tell them to reduce the water, because when the temperature is reduced, automatically shrimp metabolic activity will be reduced. So there won't be much problem with the biting of the animals, like cannibalism, it won't be there. So ammonia problem won't be there. So automatically we can stock peacefully. So this is the traditional stocking method, which one I am showing to you. So according to the temperature, the traditional method. So this is the important section which one I want to discuss with you people, the young minds. This is a nursery system, which one is adapting now in India very fastly. Almost all the maximum of the farmers, small scale farmers, bigger scale farmers, everybody is adapting. What is this nursery? This will be reducing the culture period from the growth system. So you see, so we have to make the one basement with concrete. We have to make one basement, big basement. We have to like a greenhouse. We have to install everything. So it's
just a second, sir. Sorry to interfere.
Yeah.
The participants try to avoid these things. See, this is an intellectuals presenting their presentation. So if you disturb in this way, this is not the way to behave in this forum. Kindly avoid scribbling on the screen.
Okay, sir. Thank you.
Thank you, sir.
So in this system, we have installed two HDPE tanks, which one is capacity of 50,000 metric tons capacity water, two tanks, which one it will be folded. I'll show you next slides. So we are going to culture the shrimps for 30 days in this particular nursery system. Okay. So here you can see after installing this, the aeration system is important because we are going to stock a high stocking density in this particular section. You can see we have to check the aeration. So like that, we have to check the aeration. The entire area will be getting aerated. So like that, we have to check. Okay. So this is the tank after because the tank capacity. 50,000 liters capacity, we can able to fill more than 1.7 meter depth. So we are filling up to 1.5 meter depth. We are filling the water. So this is the tank. Okay. You can see. So through compressor, we are giving the aeration because when we are doing nursery culture, if you are not aerating properly, the animal will get die. So once you are purchasing the seed, the purchasing seeds will be acclimatized and will be stopped directly to the nursery system. That we will be maintaining the nursery system. That means for 4 ponds, 5 ponds, 4 to 5 ponds seeds will be stocked in one tank. That means the density of the tank will be more, not like normal condition. So you can see here the packed seeds will be acclimatized. Here this is acclimatized tank here. So here after acclimatization through the pipe, we will be transferring to the culture nursery tank. You can see how they are feeding the feeding system. You can see. So like that. So they will be spreading the feeds the entire round. They will be spreading the entire round. So coming to the so this is the seeds after 30 days. So you can see after 30 days the size of the seed around 1 gram. So the size will be around 2.5 to 3 centimeter. You can see the seeds. So there is a uniform size. So there is no size variation. There is no size variation. Uniform size. So next come to the important part, aeration, because when you are going to transfer the seed to the culture system, grow pond, so we have to be careful about the aeration system because we are giving very good aeration system. If coming to the grow pond, if you are not giving the aeration properly, automatically the growth will be struggled. So we have to be very careful about the aeration system. So in the market there are several aerators there like pedal aerator, spiral aerators, because which one is we are recommending the spiral aerators because these aerators will be sprinkling the water. Every water molecules will be absorbing the atmospheric air and fall into the water. Automatically the water dissolution level will be increasing. So there is different aerators is there, but this aerator is very good in the market. So many people are using. So like this, you see the long arm aerators, long arm pedal aerators. So what is the importance of the aerator? Because the aerator not only increasing the dissolved oxygen in the water column, and also it's cleaning the pond bottom. Okay, so when the dissolved oxygen level is low in the pond, automatically the shrimps will come to the side. You can see the shrimps. Still, if you are not providing the increasing the oxygen level, increasing the aerator, or sometimes we can use the hydrogen sulfate. So DO powder is there, the commercially available DO tablets is there. We have to apply immediately. Then only it will be going inside, getting normal. So I already told the aerator not only giving the aeration and also it's cleaning the pond bottom. It's whatever the feed waste is there, animal waste there, everything will be settled in the pond bottom center area. Okay, so purpose of the aeration discuss oxygen supply, maintaining the water mixing, and also cleaning the pond bottom. So regarding coming to the feed management, the stability of the feed If the feed is good, the feed stability must be good. It should be good smell, attractability. The nutritive value of the feed must be good, and also feed conversion ratio must be good, and also particle size and uniformity. The size of the feed size should be uniform size. Should not be one size is small, one is big. Should not be like that, because based on the feed animal mouth size, we are giving the feeding. So that time the feed must be good. Okay, so what is the role of feed? The feed will be give the faster growth as a good FCR. It should not pollute the bond bottom and also reduce the culture days. So like that, it will be give the less production cost. So we got this is the type of feeding. So using the boat feeding, they will be spreading the feed the entire pond. Only is bun feeding, that is traditional feeding. Nowadays nobody is following. What is advanced feeding methodology, which I am going to show next? This is the advanced methodology of feeding. So for this, there is no need any manpower. So only we have to fill the pond in the machine. The machine will be automatically, if you fix the timing, automatically it will on. It will spread the feed in a particular area. You can see, you can closely watch the video. You see, it is spreading. So this is the video after spreading. So what will happen? The animal is eating or not eating. So we have the underwater camera. We went inside the pond. We have seen underwater camera. You can see the animal is capturing the feed like anything. You see, this is all the video. Everything is nothing is downloaded from internet. It's all from real. You can see all the animals is moving towards the feed. So coming to the check tray part. So what is the purpose of check tray? Where we have to place the check tray? We have to place the check tray from the 2 meter from the bund. From the bund, we have to place the check tray into that means minimum 2.5 meter depth. We have to place the check tray. What is the purpose of the check tray? Purpose of the check tray. So see like this. This is a catwalk. We used to call it catwalk. We can walk through here and we can check the check tray. So this is a round check tray. The technician is checking the animal. So next you can see, so check tray monitoring, what we will be checking. So any feed left over is there about the animal health, because every time we cannot dive and we cannot capture the shrimp. So we have to check the shrimp like that. The feed intake level, gut color, molting stage, any about the any molting. If molting is there, means we have to reduce the feed. It won't eat the feed. We should not feed unnecessarily, because 1 gram of feed, it will create 8 grams of black soil. So we have to be very careful about the black soil. Okay. So uniformity, about the feed quality is good or not, then we have to check everything. So this is a check tray adjustment. If they're not eating, we have to reduce the feed. If it is fully consumed, we have to increase 10% of the feeding. Like that is a small calculation. So so sampling. So we have to sample the shrimps. After 40th day, we have to compulsorily sample the. We have to capture the animals through the cast net. We have to weigh the animals. We have to check the average body weight of the animal. So you see, like that. So we have to during the sampling, we can check the animal health. Any necrosis is there, any weight cut is there, any muscle cut is there, any problems is there. So like that, we can be able to check. So I already told during the aeration, the center of the pond, all the waste will be accumulated. So what we have to do, we have to purchase a submersible motor. In the center of the portion, we can put it and we can release we can eradicate the unwanted, the dark soil, water, the soil. We can disturb and we can remove it, because it will be give the better. It will be the better water quality management. Okay. Still, people are struggling. I don't know why. The goal, so we have to reach the better growth.
Excuse me, sir. Sorry,
yeah.
I'll just make changes and I'll remove this.
Okay, so the harvest is the important chapter, because you are preparing pond preparation properly. seed, you are selecting good seed, probiotic application, you have cultured the shrimp. After that, you have to sell into the market. Then only you can able to get your profit. So you can see the people are capturing, and also they are carrying to that. They will be killing in the ice. We have to kill with the ice. So what their recent technology is telling, previously we used to put the crusher ice. So now they are telling don't put the crusher ice. For the better meat quality, you can put the large ice cubes. So after that we have to after killing we have to put it like that. The buyers will be buying the materials. So many buyers is there in India. So we have to see the hand picking. So we have to do the hand pick. 1 or 2 animals. Some animals they won't. It will be going somewhere. That means pond area is more now. So it will be spreading somewhere. So we have to do the hand picking. So some of the problems we have encountered to the entire crop. Some cannibalism will be occur. Sometimes the cannibalism will be occur. Sometimes the size variation will be there. You can see some coloration of the animal. See some protozoal disease. You can see some looser. The animal will be, it won't be tight. It will be very loose. So that means the nutrients of the feed is not there. Some microbial problem is there. For the protozoal disease, it's a protozoal problem. For the coloration, this is a mineral problem, mineral deficiency. For size variation, that improper seed quality. So this improper seed quality, cannibalism because of animals that stronger animal will biting the weaker animals. Okay, what is this? This is the gastropods. Because when you are not properly filtering the water, the gastropods egg will be passing through the directly, that maybe some damage of the filter bag, automatically eggs will be fall into the water. So they are establishing the community. So we have to eradicate it. Otherwise the feeding FCR feed conversion ratio will be more. So these are the recent problems. Which one is in Lithuania is Vanami, white cud and white fecal problem. This is a major problem in the industry. Because of that, the animal is getting harvested by 10 grams or 15 grams like that. After once the white cud, if you are not properly managed, properly cured, we have to harvest the shrimps. So these are the white mussel disease. This is due to mineral deficiency. The disease coming. So lack of mineral deficiency, because we are stocking high stocking density, like 50 pieces meter squared, 70 meters per meter squared, like that we are stocking. So we have to be very careful about the mineral in the water column and also nutrient in the water column. So we have to often we have to use magnesium chloride and potassium chloride. So if the pH, alkalinity, nitrate, so we can have the running mortality. You can see the check tray. So plenty of animals is died. So that is running mortality syndrome. Later they have come to the conclusion, because the daily mortality syndrome or running mortality syndrome on our funds. So which one as I'm telling, this is the white spot syndrome virus. So the disease came by year 2005. Still there is no medicine. So many projects, so many people, they are keep on trying, but they cannot able to control, or there is no proper medicine to the disease. Once the disease get affected, ultimately within a day, if you are not harvest, all the shrimp will be died and it will be floating like this. This is because of the over blooming. So this is the black gill, because the water, once the water is getting very thick, automatically the gills also will get cracked. So some of the other problems, like we have to side because of the algal deposition, algal plankton crash, plankton death. So it will come to the side if you are not removed properly. So like this, it will become the black side. So it will be create the problem. So we have to remove the maintain the pond is very, very important. This is cannibalism. So these are the feeds that we are using for the Vanami as well as modern, based on the shrimp mouth size, different number of feed size is there. So now I am coming to the particular topic, scope and opportunity. So what are the, maybe the people might think after MSc Zoology or Science, what we will do? So you have a lot of chance to go, like a farm technician or farm manager or consultant. Consultant means maybe after 5 years, 6 years you are working in the farm, so you can promote it as a consultant. And also so many chemical companies is there. So more than 170 companies is there. feed mill. The feed mill. So many feed companies is there. More than 15 companies is there in India. So you can join in the feed companies like marketing executive, the technical manager, like that. The nutrition advisor, you can be a nutrition advisor. So lab. So for the checking the water quality, so many labs is there checking the viral problems. So PCR laboratory is there. Hatchery you can join. So if you see the employment opportunity in aquaculture, the online on-farm jobs like production, processing, marketing. So after selling all the shrimp, dead shrimps will be go to the processing plant. There they will be processing. So there you can get the job. So if you come to the academic, you can be a research teaching, researcher, technology. If you go to the government, you can be a administrator or a production manager. MPDA you can go, CMFR you can go, CFI you can go, Central Institute of Fishery Education, Central Institute of Brackish Aquaculture, then Central Institute of Fishery Research Institute, Marine Product Export Development Authority. So like that, so many government organizations is there to state government of fisheries can join. So like that you can go for you can sell the shrimps, you can purchase the shrimps. So like that also you can start your own own business. So not only that, some of the people is very much interested on satellite system. So you can make your own device. Then you can help the farmers to automatic reader of when the DO level is decreased, automatically you can make a call. So like that device also is there. So using the remote sensing, the technology, they are creating so many models. So this is the this is sustainable production consumption. So for input, then production, then processing, then marketing, then quality and safety. So this is the sustainable production and consumption. So everywhere you have the job opportunity, from starting from the input, ending from the quality and safety. You can have the job opportunity. The thing is, we have to check the opportunity, because in India, you can see 65% of the production from aquaculture only. So you can see the marine and inland and total. So you can see that how much gap is there to fill. The inland, how much gap we can culture plenty of shrimps. 74.18% gap is there. We can fill the gap. So 170 hectare, 170 up to 170 level, that hectares is there to develop. So this is the concept for the successful shrimp farming. So if you have the clean pond, clean seed, clean water, nursery, probiotic farming program, scientific farm management, complete biosecurity, so you can win the match, you can win the game, and you can get a profit. So this is the take-home message from my side. So if you have a good seed, genetic seed, excellent probiotic farm management, quality feed and inputs, timely harvest also important. If you're not harvest timingly, so it will you might lose your money, because you have to see when the market rate is increasing. So you have to look after like gold rate. So we have to look after when the shrimp rate is increasing, then you have to look after the rate. So automatically you can win the successful culture. So this is the conclusion point. There is no medicine for virus disease so far. So prevention is always better than cure. Culture industry could be sustainable if everyone is sincerely join hands and work together behind your smiles. So great achievements. Thank you for your patience. If you have any queries, conclude.
Any questions, clarifications?
Kunal, sir, I have a question, sir.
Yes, sir. Please tell me, sir.
Yes, sir. Sir, it was a really informative lecture.
Yeah, tell me.
So you mentioned that for white spot syndrome, there is no medicine as such.
Yes, sir. Yes, sir.
But I mean, I have also worked with white spot syndrome virus in the case of prawn.
Okay.
And then I came across some literature on DNA vaccine.
Okay.
Does it work in the field, sir?
Actually, sir, in Thailand, sir, when I visited in the year 2000, Thailand, that time itself they have started because I'm working with CP Aquaculture India Private Limited. I have visited many times Thailand and research laboratory. So that
Yes, sir.
time they have come with the Semvac, then some powder, vaccine powder. But
Yes,
thing
sir.
is, it's not working out because even last year also plenty of farmers got affected by white spots virus, especially with the Japanese one. Maybe in the laboratory condition, it might be working, but the field we could not able to get the results. Sir. That is the truth.
Okay, sir. Okay.
Yes, sir.
Thank you, sir.
Thank you, sir.
Any other clarifications? Sir, name some probiotics used as feed for shrimp.
Actually, sir, if we tell this things, sir, like means we are recommending some probiotic means it's not good actually. So mainly, basically for probiotic, that combination of the baseless species combination is good for the water quality. For the soil probiotic, we have to use the rhodococcus and rhodofactor, which one is having that it will give the good result. Rhodococcus and rhodofactor bacteria will give the good result. And also for the feed mixing, same baseless species, but we have to give it a good concentration. The cells per unit will be should be more. So these are the generals, sir. Because market so many companies is there, sir. In aquaculture, more than let's say more than 170 to 200 companies. Maybe if I am working with the company for 5 years, next after coming out from the company, I can start my own company. So like that, people are doing. Yes, sir.
Is there any live feed provided for the shrimp culture, sir?
Sir, actually, live feed, no, sir. Actually, in hatchery, they are giving Artemia when there is a Zoea, mysis, that stage they are using the live feed. But in the case of culture, nursery system, we have high nutrient feed, artificial feed, as well as in shrimp culture grow ponds also artificial feed only. We are not actually previously when they are do culture on Pinus monodon, that time they used to they have the habit of using clam meat in the end of the culture. Not always. End of the culture they used to have the clam meat. But nowadays nobody using because the clam meat, using clam meat, it will pollute the pond bottom. So next culture maybe it will be giving very big trouble.
How long does it take, sir, the whole process?
Actually, for the culture period is 120 days, sir. If you have the nursery system for 30 days, you can able to culture within 60 to 70 days in the growth system, sir. Because if you are using nursery system, what will happen? The importance of nursery system, automatically the good seed will be survived. Because maybe if you are stocking some 3 lakh seed in 50,000 liter capacity of water tank, what will happen? Automatically you will get some 70 to 75% of the survival only. The remaining 25% will be die because when the suitors will be surveying the nursery system. So when the suitor is surveying nursery system, after transferring to the grow out, automatically it will grow fast. It will be jumping growth.
What is the importance of seed genesis for shrimp culture?
What are the
seed genesis for shrimp culture? Seed genesis. Seed genesis for shrimp culture.
Seed genesis. I don't know.
Something
Sir.
they have. Okay,
Yeah,
okay. Sir, baby can baby.
Actually, sir, since that Lithopenes vanami is an exotic species, the government is very strict on that policy. They are using specific pathogen-free broodstock, specific pathogen-free seeds. Okay, that is turbo seeds. They used to say turbo seeds. That seeds only they are giving to the farmers.
Sir, can baby brine strips be also used?
What's
baby brine shrimps can be used. Also used.
Yes, a baby brain shrimps can be used for the hatchery system, because hatchery they are using artemia. When we are, because why we cannot be able to use artemia or baby brain shrimp is artemia. So why we cannot be able to use, because the rate will be higher, because the production cost will be increased like anything, because now itself, because the rate now, so for the 50 count now the rate is 330. So 50 counts means the shrimp 20 grams shrimps 330 rupees. Okay, if the shrimp is 25 grams, the rate is around 400. So like that now, so we cannot able to get the profit. So we are doing culture for profit, not for jolly something. So we are, when we are doing culture, so much of effort you are putting, so high risk you are putting, so you have to gain the profit.
Termites can be used as feed, sir. Termites.
Termites. No, sir. For the, actually, for the brooder, they are giving polychaetes. For the brood stocks, now they are maintaining that SPF brood stock. For the brood stock, there is a specific checked polychaetes. The polychaete checked means they are checking polychaetes also white spot syndrome virus, because polychaetes also can carry the white spot, because they are bringing from the brood stock from Florida. Here they're bringing here, after feeding, through the feeding they should not get affected. So they are very keen on that.
Is there any fund released by government for the shrimp culture, sir?
Actually, if you have if you want to culture the fish farm, government giving subsidy, some 50,000 rupees something they are giving subsidy. If you are approaching the state fishery office, they will be giving the idea regarding subsidy. But shrimp farming, they are giving subsidy for, if you are a big farm, they will give you subsidy for creating your own laboratory, for processing plant. This thing and all government is supporting, MBDA is supporting, Product Import Export Development Authority is supporting, and also National Fisheries Development Board. They are nowadays they are aqua cleaning for aqua entrepreneurs. So there is a like a training program they are conducting. Those who are undergoing the training program, they are giving some 7 lakh, 8 lakh rupee for creating their own laboratory or developing their products.
Can this method be applied to breed ornamental shrimps at a smaller scale?
Ornamental, actually ornamental shrimps, there is two, only is marine ornamentals, one is freshwater ornamentals. Ornamental shrimp actually, let's say one of my colleague actually is my senior when I studied college. He having is Dr. Saudi Arabia. He having his own ornamental hatchery in Rameshwaram, Mandapam area. So if they are interested, I'll give the phone number, sir. They can contact my mail. I'll give the phone numbers.
So type your mail there, sir, in the chat box so that they can contact you, sir.
Okay, sir.
Chat box, you can.
Yes, sir. Sure.
So one last question, sir.
Yeah, tell me.
Live feeds are good for shrimp culture. Is this food enough?
Yeah, live feeds are good enough to give better growth. Like I told, zooplankton, so initial growth will be boosted. But the thing is, we cannot be able to always give the live feed, because the cost of production will be increasing like anything. Okay.
Okay. Your mail ID on the chat box.
Yes, I will share, sir.
So thank you, sir. Ravi Kumar, sir.
Sorry, Shiva.
Yes, sir. Your concluding remarks.
Good evening, one and all. First of all, I thank the Gunalan sir for accepting our invitation during this COVID time and spending your valuable time, sir. Your talk on shrimp culture techniques and its scope was really excellent. You started with why to know about shrimp culture, and you started with the history of shrimp culture, and you gave many ideas. In that, you gave one thing about WSS, the outbreak. I think it will give an idea for students who are want to pursue research. Also, I touched upon blue economy, and you said something about biosecurity, that is bird and crab fencing, and the use of probiotic applications. You also gave the complete insight about shrimp culture and its techniques. I think it has motivated many students. And once again, I thank you for sharing your vast experience with
Thank you,
me.
sir. Thank you.
And I'm really sorry for the inconvenience caused in between.
No problem, sir. No problem. No problem, sir. No problem, sir.
Thank you, sir.
Okay, sir. Thank you
Thank
so
you
much,
so
sir.
much, sir.
Thank you, sir.
Thank you so much. So, dear participants, now we'll have a game. That is, we'll continue the game for this session. So, I request everyone to join. I'll share my screen. So this is the code 487644. Now we have to just join the quiz. You need not register as you have registered already. Just you need to join the quiz.
So thank you so much, sir. Can I leave, sir?
Thank you, sir. Yes, sir. Thank you, sir. Thank you so much, sir.
Thank you. Thank you for your giving the opportunity, sir. Dr. Tyagarajan, sir, and Dr. Sivachandran, sir, and my good friend, Dr. Rajagopal, sir, and also the principal, sir, for allowing me to present here. Thank you so much. Thank you, sir.
Thank you, sir. Thank you, sir.
Thank you, sir.
So we are starting the game. Participants, we will be starting the game. And once you complete your quiz, you can go for your feedback. I'll be posting the feedback on chat box. So we are starting the game.
Shiva. Shiva. Hello. Shiva. Shiva.
Sir.
Feedback link.
Sir.
Feedback link எதில அனுப்புவீங்க?
போட்டாச்சு. குரூப்ல போட்டேன் பாருங்க.
ஓகே சார், நிறைய பேர் கேக்குறாங்க, அதான் கேட்டேன்.
போட்டுட்டேன்.
ஓகே.
3:24. எல்லாம் ரெஜிஸ்டர் பண்ணி வச்சிருப்பாங்கப்பா. இது போட்ட உடனே டக்குன்னு ஆஃப் பண்ணிட்டாங்க. So first place goes to Sibu, second Manjita Ghosh, and third is unsafe name. See the accuracy of the game, only 61%. The first session it was good, and the second session class accuracy is just 61%. So you need to be more attentive participants. So thank you all for today's session. 2 sessions. I thank all the invited speakers and the participants. And here we end the session with this quiz, and hope everyone has filled in the feedback form. The feedback form will be there for some more time. And tomorrow, exactly at 3, we'll start the day 2 session, topic sericulture. And I expect all to participate in this. And thanks for your cooperation. And I request participants not to scribble anything on the screen, as we should respect the speakers for their valuable work they are doing and they are presenting here. So I request all the participants to make sure that you behave well, as this is only for students. The main target audience are students. So you have to benefit out of it and not make anyone embarrassed. So kindly request you to maintain the decorum and discipline while you are attending the program, and hope you will do so. Thank you all for the participation, and the feedback link will be there for everyone, for the participants, and you can fill it. By 5:15 we'll be closing this. No, 5:30 we'll be closing. Hope you are enjoying the quiz, and every day we'll have a quiz at the end of this session. Any doubts, any queries, participants can raise any clarification. They can unmute themselves and get clarified. Any clarification tomorrow, mail will be sent by evening or tomorrow morning. இப்போ போட்டிருக்கேன் பாருங்க சார்.
Okay, sir. Wonderful, sir.
Thank you, sir.
Thank you. So, sir, okay.
Sir, சார் கொஞ்சம் மியூட் பண்ணிக்கோங்க சார். சவுண்ட்
Okay, sir. Okay, sir.
எக்கோ வருது. Participants, if need any clarifications or any information, you can unmute yourself and you can ask. Those who have filled the feedback form can.
Excuse me, sir.
Yeah, yeah.
Sir, the e-certificate, is it combined for all the courses?
Yeah. For all the
close till I get up. Time is barely on our side. I don't wanna waste what's left. The storms we chase are